In this video, we will discuss about this how IMPDH2 downregulation leads to the DNA damage accumulation and also this cell cycle arrest at G2 and M phase. So in the previous video, we have understood that IMPDH2 has a role towards this prevention of DNA damage in the triple negative breast cancer cells. So they wanted to ensure that this IMPDH2 has a direct role towards this DNA damage accumulation, or not a direct role. This IMPDH2 has a direct role towards this DNA damage repair. Yeah. So first, what they did is they checked the cells. They checked this IMPDH2 locations in the cells at this using their in-house developed cell cycle tracking assay. And what they have found out is that this nuclear IMPDH2 deviates to some extent in the top 10 percentage of the cells that is seen in the panel A, left hand side of the panel A, but in the bottom 10% they can see the some of the accumulations is higher. Some of the accumulations are more in this G1 and G0 and G1, G0 and G2 phase, but no cells were found in this G2 and M phase. Thereafter, they check the nuclear localizations in different kind of in different cell system and different stage of the cell cycle. In the panel B, here they have find out is that the G2 and M phase there are more IMPDH2 are found compared to the IMPDH2 in G1 and S phase. And these things they have also validated using the immunostaining, where they have seen that in the turquoise 2 the intensity of IMPDH2 is lower compared to the case of the cells that are found in G2 phase, that is turquoise 2 G2 phase. In turquoise 2, here we can see that intensity is high compared to the IMPDH2 that are found in the G1 phase. And these things are also quantified, and here they have quantified the markers for the DNA damage, and what they have find out is that DNA damage is higher in the case of G2 and M phase compared to the other phase. This indicates that cell cycle arrest are occurring. Cell cycle arrest are usually occurs in the phase of this G2 and M, and where the DNA damage is accumulated. And these things are also in line with the literature, where it is shown that when the cell cycle moves from this when the cell cycle moves to the S phase, it cross checks whether there is any DNA damage and repair. So they have found that in the literatures that cross checking occurs when the cell cycle is about to move to S phase. So it will stuck up in the G2 and M phase, and because the DNA damage is continues to rise because of the absence of IMPDH2. Thereafter, then they wanted to. So in this, so far in the panel A, B, and CD, they do not knock down IMPDH2. So it is just the inherent characteristics of the pancreatic cancer cells, not pancreatic cancer cells, breast cancer cells, that has more DNA damage properties. Now they wanted to check whether it's the IMPDH2 that cause this DNA damage. So what it is, they knock down, they silence this IMPDH2 using silencing RNA. What they have find out, there is an increase in the amount of markers. There is an increase in the signals of the markers for the DNA damage, that is H2AX, for both the cases compared to the control case. These things they have observed for MDA MB231, that is triple negative breast cancer cells, and the Cal51, another breast cancer cells. Similar results are observed. and these things are quantified in the panel F, where we can see that uh, with the increase in the with the presence of the silencing RNA, the amount of DNA damage is high, is statistically it is statistically significant compared to the control case for G1 phase, S phase, and G2M phase. And when after confirming that the silencing IMPDH2 will cause more DNA damage, they again checked it using different approach where they knocked down IMPDH2 using CRISPR-Cas9, and thereafter they check the DNA damage. They check the DNA damage. So right now they are not knocked down in the panel G. In the panel G, what they did is inhibit they inhibit this IMPDH2. They wanted to check whether it's the activity of IMPDH2 that cause DNA damage or not. So they inhibit this IMPDH2 using MPA or using MPA in a dose dependent manner. Dose dependent results are in supplemental figures. So in this figure, what they did is they use the inhibitors for IMPDH2 and check whether it will cause or DNA damage or not. So they can see the DNA damage, and it's comparable to the control where that comparable to the control that is toposidase that is known to form DNA damage. And these things are also quantified in the panel H, where we can see that with the dose dependent injection of this IMPDH2 inhibitor, that is MPA, there is an increase in the DNA damage marker signals in the nucleus. And this thing they have observed for G1 phase, S phase, and G2M phase. And once validating that it's the activity of IMPDH2 that is responsible for DNA damage repair, now they wanted to check whether if this IMPDH2 has been completely knocked down, whether it will cause uh, the accumulation of DNA damage or not. And what they have find out is that when in the case of IMPDH2 knockdown, they see increase in the cell damage markers and other proteins, that is 53BP1 also, which is also involved in the with this DNA damage process. And thereafter they also wanted to see whether if this IMPDH2 has been introduced in the knockout cells, whether it will reduce the DNA damage or not. So they introduced this guanosinine 50 micromolar that can basically plays a role towards roles as an alternative to IMPDH2, and they wanted to see whether it will cause some suppressions in the DNA damage or not. And from the immunostaining figures, it is not clear, but when they quantified, they are able to see that there is some decrease in the DNA damage in the knockout. There is some decrease in the DNA damage in the knockout. However, it has a negative role on the wild type because of the unbalance of the guanosine in the nucleus. And when they check the 53 other markers for the DNA damage, that is 53 pp1, they see that with increase in the guanosine, those markers also goes down. That is at 0 guanosine micromolar, the markers of this DNA damage, that is 53 pp1, is high, whereas for the 1000 micromolar, the markers for this 53 p1 is lower. And then they wanted to check whether cells will again acquire its property or not. So they check the knockout IMPDH2 knockout proliferation properties with the cells that has the wild type IMPDH2, and they are able to see that the to some extent the knockout cells are able to regain the properties, but at the same time for the wild type their properties goes down, and this also the authors attributed to the unbalance of the guanosine within the cells. So from this they made their claim that IMPDH2 is directly related towards DNA damage repair, and IMP and its catalytic activity is and it is categorically active towards repairing this DNA damage. The downregulation of this IMPDH2 will cause DNA damage and cell cycle arrest at G2 and M phase. So why G2 and M phase? Because most of the this upregulations of localizations of this IMPDH2 is found more in the G2 and this G2 and M phase compared to the S phase and G1 phase that can be seen here. It's higher in the G2 and M phase for all the results that are shown here, and other results are also shown in the supplemental figures. Here also we can see that it's higher for the G2 and M phase, and in the panel A we can see that most of the cell cycles, some of the cell cycles are this bottom 10% of the cell cycles, bottom 10% of these cells are in the this bottom IMPDH2 cells are mainly in this G1 and S phase, but high nuclear IMPDH2 cells are in this G2 and G2M G2 phase. So that's all for this video. If you want to understand more about these experiments in detail, you can go to the reference that is provided in the description. You might want to refer to the paper to understand about this G2 and M phase. Those results are more clearly or unambiguously presented in the supplemental figures. So that's all for this video. Thank you for your time.